Large to small horse you see here, there are isolated species, and we have no way of proving that one actually became the other, except by inference. Remember, all of these theories lie at the end of a long inferential trail. No one saw this happen. A second thing Darwin couldn't know, and what we'll concentrate on tonight, is the degree of complexity that occurs inside the cell. Darwin and his contemporaries thought that the cell was just a living blob or protoplasm, not really worthy of study, just like little cubes of jello that were alive. And we know that that's very different now. This is a picture of Michael Behe, who's one of the leading proponents of this thinking. He's a professor at Lehigh University in Pennsylvania. So naturalism, Darwin's theory, I'll use Darwinism and naturalism interchangeably, would state something like this, that life arose by undirected processes, and at given time, the improbable becomes possible. Time plus chance are sufficient causes to explain the origins of life. And we're going to look at the boundary of what time plus chance can actually achieve. However, all the college and high school textbooks teach this cosmology for the origins of life. And I'll just review it briefly because this will help us understand where we go next. In the primordial earth, and somewhere on its surface, is something called the prebiotic soup. Prebiotic means before life. So before there was any life, life had to start. And on planet Earth, in a sheltered pool of water somewhere, there would be this prebiotic soup which consists of the building blocks of life. Now, the building blocks of life are something called amino acids, and they work kind of like a child's building blocks, and we string them together like the cars of a train. You start with the locomotive, the coal car, the caboose, and they'd be strung in a length that creates the functional proteins that allow the cell to work. These are the basic building blocks of life. So in a prebiotic soup, you've got the individual pieces, the amino acids, floating around together, mixing randomly. Remember, nature with a small n, no direction, just random assembling of these small groups of proteins, small groups of amino acids, like as you see here. Eventually, these smaller groupings become longer, and as these amino acids are lined up side to side, they each have chemical properties that give by their sequence, that is, the order in which they're put together, a three-dimensional function, shape, and form to a protein. Now, the proteins are very long. They can be 50, 100, up to 500 and more in length. But you see a picture here of what it would be like for the building blocks of life, the amino acids, to assemble themselves by chance in this primordial soup. And it would look something like this. This is an amino acid shown in three-dimensional relief. Let's imagine that one of these atoms up here that you see in schematic form as one of the building blocks of life, an amino acid, was the size of a tennis ball. I want to give you an idea of how complex a cell is and how big it is in relation to our atomic structure. So if that's one of the building blocks, an amino acid, that we have to string together, hundreds of them in length, to build a three-dimensional protein, if one of those atoms was the size of this tennis ball, the amino acid would be about that big, and an enzyme or a protein that does something would be about the size of a Volkswagen or a truck. Okay? Now, if one of those enzymes, that is, the functional units that make our body work, are the size of a car or a truck, how big in scale would one of the cells, the trillions of cells in your body, be? Well, let's see. Let's take this tennis ball, and we're going to translate it into the cell. And it turns out that if an atom is about the size of this tennis ball, one of the cells in your body is about the size of Manhattan. And the interesting thing about it, and something Darwin could not know, what he considered as just a living blob or protoplasm, a piece of living jello not worthy of study, actually is as complex or more so than Manhattan. There are docks that exchange information with other cells. There are power stations. There are chemical plants, there are factories, there is a library, there is a central government, there is a telephone network, there are subways, there's continuous construction going on, and frenetic activity at the molecular level all the time. Let's do a mind experiment and suppose that I was going to send a message to a cell in your body and ask the cell to produce a protein that is an antibody that functions to fight infection. So I want to send a message to a cell, and I want it to make this protein for me. This is something that our body asks these individual cells to do. It looks something like this. Let's imagine now that the cell is underneath us there, and I send a message to the toll booth. It gets through the cell wall, and it's translated to a second messenger, which takes it by taxicab, and this message gives the taxicab the message of where it needs to go. It says, I need to go to the nucleus because I have to get to the central archives, the blueprint for the entire cell, and I need to copy a part of that blueprint for the antibody I need to go to. That taxicab will then drive past all the construction, past the power stations, past the sewage plant, past the entrance to the subway, finally arriving at the gates of the nucleus or the archives where the blueprints are kept. And when we pass into the nucleus, we see the DNA molecule, a single length, 5 billion units long, that has the blueprints for the cell. The taxi, having been given the correct message, goes to just that point of the DNA that has the information I need to make that antibody. A third messenger is then created, sent out into the cytoplasm to find the right factory where the raw materials exist to make that protein. The protein is then processed by a second protein that's already ready to go. And then it's attached to a truck, and the truck takes it back out of the cell so that it can do its job. Darwin couldn't know any of that was happening. He had no idea. This is nanotechnology on a fantastic scale. This is what they're trying to do, and it already exists in every cell in your body. Biological machines, we truly are wonderful. So let's consider just one of those biological machines. When our second messenger went and attached itself to the DNA, it needed this enzyme called a ribonuclease. What the ribonuclease does is it allows translation of that piece of the DNA or blueprint for your body and translates it into another chemical message that the factory can read. You need this ribonuclease for life. You cannot have life that reproduces itself without this enzyme. It's about 100 units in length. Okay? You've got to have it. This is part of an essential for any kind of life. And let's look at what the chances are of this ribonuclease occurring. Now, one of the hallmarks of design, I promised you that I'd talk about how we can tell design from non-design. One of the hallmarks of design is something called complex and specific information. I'll illustrate like this. Imagine you've got the prebiotic soup now with the building blocks of life. I'm going to represent them with kind of like a Scrabble letters, because there are 22 biologically active amino acids in your body. It's kind of like having an alphabet soup. Let's suppose we got the alphabet soup, the amino acids are floating around, and I'm going to try to create the correct sequence from our Scrabble pieces. And I'm going to get some help, okay, in our experiment. And we have Storm, the amazing Scrabble-playing dog. So he's pretty good with that, but he's very close to chance. So we're going to give Storm a chance to play with our Scrabble and see if he can come up with the correct sequence that we're looking for. Now, if I put Storm in the prebiotic soup and let him come up with this sequence, how many of you think that Storm might have done this? Is this an example of complex and specific information, which is a hallmark of design? Well, you can see there's 3 letters together. It's very specific, T-H-E. How many of you think that Storm could have come up with this sequence? Now, what if Storm made his prebiotic soup into this sequence? What would you think? Well, it turns out we can actually measure the probability of our prebiotic soup or Storm coming up with this by chance. Let's look at this statement is an accident. It's a 29-member sequence, and there are 27 degrees of freedom, that is, at each part of the sequence. It could be any of 26 letters or a space. So Storm goes down the line and picks a letter or a space for each one of the sequences 29 times. The probability looks something like this. 1 over 27 to the 29th power. Well, this number doesn't mean anything to me. I don't know if it means anything to you. But it's like really small. How small is it? Well, suppose we go to California and you play the Mega Millions lottery. Okay, I don't know if there's any winners here, but you'd have to win this lottery. She's a very happy lady. She's got the big check because she's found this statement is an accident. But the problem is, not only do you have to win the lottery, you have to win it 5 times in a row. If you won the lottery 5 times in a row, that's equal to the chance that Storm could come up with this statement as an accident. So we underestimate the degree of difficulty there is at arriving at a proper sequence by chance. But then you say, oh, Carrie, what if you have a lot of time? You're underestimating the power of time if we let this go on and say, okay, let's look at this. These numbers are so huge that I was staggered when I first ran them. And that is, if we took the Milky Way galaxy, 100 billion stars, that's a lot of stuff, and we turn every particle in the Milky Way galaxy into a building block of life, that amino acid from the prebiotic soup, turn the whole Milky Way galaxy into substrate that is the raw material for life. And we allow those pieces to interact together 1 million times per second, which is close to what they really do. Giant prebiotic soup, and give you 10 billion years to shake it up and let them randomly associate into 100 sequence-long chains. What are the chances that we would come up with this ribonuclease? 3 times 10 to the 104th power. What does that mean? It means that you've got to be really lucky because we're going to win the lottery again. You're a winner 14 times. Now, this is different than before because it's not that we're waiting to win 14 times in a row. This is saying for the one protein to have occurred by chance, given the entire Milky Way to operate in, you get one chance to win 14 times in a row. And it really tells us there is no chance at all. Somebody's cheating. And it's interesting that you can't get anywhere anyways with one ribonuclease, because doing something as simple as metabolizing sugar, which all organisms must do, everybody's got to eat, requires a whole set of enzymes. The body doesn't do anything simply or easily. It's always really complicated, which is why in biochemistry it drives you nuts. Because this is how we metabolize sugar. The blue letters up there, each blue word represents another protein at least as big as that ribonuclease. In order for you to digest the sugar you had for breakfast this morning and you put in your coffee, you need all these enzymes, and this pathway is what's operative to metabolize that sugar. It occurs all together.